Okay, good afternoon, everybody. It's my great pleasure to introduce our guest speaker today, so Jean-Philippe Vert from Google Research in Paris, and also NIM Paris Tech Center for Computational Biology. So I don't have to really introduce him because he's a quite well-known researcher in the machine learning community. But now, so he's at Google and also the university. And previously, so he was a postdoc actually in computational biology at Kyoto University. So that's 2001 and 2, so about 20 years ago. But then after that, actually some of the members of AIP have nice collaboration with him. And also I'm one of the members who are influenced by his research area. So it's my great honor to introduce him today. So today he will be talking about deep learning for biological sequences. So I think so. Today the seminar is done in a hybrid mode. I hope my voice and his voice can be heard clearly. If there are some problems, please let us know. Okay, then Jean-Philippe, to start your talk.
Thank you. Thanks a lot for the kind introduction and the invitation to be here. It's a great pleasure to be back to Japan, to Tokyo, after these few years of no travel ban. And so, as Masashi was saying, I know quite a few people. I don't know if they're in the audience, but I have many colleagues and friends in Japan, so I look forward to see them. And so today, yeah, I will discuss a few things that we've been doing with my group at Google and at we call them in Paris, related to machine learning, so deep learning for sequences, but in particular for biological sequences, so in particular DNA and protein sequences. And so, there will be a mix of some biological applications here, but mostly I will focus on the machine learning aspects of it, and you will see there are a few generic problems that it happens, and there will be a few open questions that maybe you may be interested to discuss more. So maybe the context of the talk is, if you work in AI and machine learning, you probably know that one field in particular has been completely changed in recent years. It's the field of natural language processing. So when we study languages like English, Japanese, French, and there's been a real change of paradigm with the notion in particular of large language models. So most of the applications that you see that even at Google, but beyond Google, in language translation, for example, or text classification or text generation, etc., now more or less use a common framework called large language models. So here I just, this picture shows one of them called T5, but there are many more of them. T5 is just one model developed at Google that is a unified model to do many things: translation, sequence classification, summarization, etc. And basically, how the model works, it's not to talk about T5 itself, but what's interesting is that this is a model. So this particular model is a model that does sequence-to-sequence translation. So it's a model that has a sequence as input, a sequence as output, and many applications can be formulated as a sequence-to-sequence problem. And what I find interesting is that in order to do this sequence-to-sequence problem, it goes through something which is basically a continuous model. So the discrete text is transformed into a sequence of vectors, and then there are many operations. So there are transformers-based, architectures. So then there are lots of linear algebra, and it's very useful because with linear algebra we can use the large computer that we have at Google and beyond. So the GPUs, the TPUs, they are very good at matrix multiplication, etc. So it works in continuous domain. And then from the continuous domain is mapped back to discrete. Right, so maybe the only thing that I will keep here is that in order to have state-of-the-art models for text, and in particular discrete text to discrete text applications, the state-of-the-art is to go to a continuous space where text is represented as a continuous vector or matrix or tensor, and from there continuous operations are applied and allow in particular to generate text. So the basic idea that we pursue this, I will pursue this presentation is, can we follow the same strategy for other texts that are important, which are biological texts? And so I started saying there are texts in biology, including DNA. So DNA, you've heard of that. RNA, probably you heard of that if you heard about vaccines. We have RNA, messenger RNA vaccines. You can also, see them as text of letters in a small alphabet. Proteins, I will talk a little bit about proteins. They are, in fact, chemically, these are polymers, but a polymer is a sequence of basic things, and a protein is a sequence of amino acids. So there are 20 amino acids, and therefore we can represent any protein as a sequence in an alphabet of 20 letters. And so following the idea of natural language processing and T5-like model, maybe it's possible to create models where all these sequences are embedded or presented as continuous object in some space. That would be the equivalent of my T5 box. And then from that space, we would be able to solve problems which arise in biology, like sequence annotation or classification. Like if you have a new sequence in your genome, you would like to know what it does, what are the proteins, what are the functions of the proteins. We may want to do efficient search, like if you have a new genome, you want to search if there are similar genomes sequenced somewhere on Earth. You may want to predict the 3D architecture of the protein. You may want to generate. This is, I think, one of the most promising applications in that field, which is, can we create a new protein, so a new sequence that would have some functions. And so you see all these things basically are parallel to what we do with text. We also want to generate text. We also want to classify texts. And so you can imagine that these applications also are very important and meaningful in biology. So maybe you can just borrow the same ideas and apply them. So I'm not the first one to suggest this idea. Actually, it starts to be a pretty mature field. Many papers have been published already about, like here I just show a few titles, but when you see learning the language of viral versions is basically a language model on DNA. When you see improved protein structure prediction using deep learning, I mean, this is AlphaFold. This is also using deep learning for specific things. So I will not detail all of these things, but basically many teams already have said, well, we have sequences, so let's take models like transformers, CNNs, whatever we have, in order to already try to solve many of these problems using the technologies we already have. And so what I would like to discuss today, this is a research talk. I will just discuss a few aspects which I think may deserve further interest, because in fact, there are some differences between language, so between Japanese or English and DNA and proteins. And so I will just discuss a few aspects which I think are specific to biology, and see how they imply possibilities of developing new models or new ways to train the models for biology, right? And in particular, I will focus on three things. So it will depend on time. I will try to manage my time. The first, so basically the three topics I will discuss are a bit specific. So because I did work on that, but they concern three different levels in, let's say, a model for biology. So the first one is at the very bottom level. It's how do you transform a sequence into a continuous object? When we start with NLP, for those of you who have worked with language models, you know that there is a phase called tokenization that starts from a sequence of characters and then parse it into a sequence of what we call tokens, and then embeds the token, and then that's the beginning, that's the input of language models. So should we do the same with biology? It's not obvious, because the basic unit in biology may be different from basic units in text. That would be the first topic. The second topic is specifically for DNA. The fact that when we see DNA as a sequence, in fact, is an oversimplification of the truth. The truth is that DNA is a double helix. Probably you know that. And so when you take a strand of DNA, you have two helices, and so you have two texts, in fact, because each strand has a text. And so you could say, well, I just take one of them and forget the other ones. But if you remember that you have two equivalent texts for the same object, then this may suggest symmetries or invariants that you want to constrain in a model. So we'll discuss how the architecture of the model could be made so that the natural symmetry in DNA is conserved. And finally, I will discuss specific tasks. So in particular, I will discuss alignment. So there are some tasks which are super important in biology, but which are not relevant in text, in language processing, like alignment of proteins and text, and DNA. And so we will discuss a bit how these alignment tasks can be added as a loss function, if you wish, in order to train the models. All right, so this does not cover everything, but at least it's I think, some illustration of what is specific to biology and how this translates into computational models. All right, so let's start with the beginning. I said the first step is you have a protein or DNA sequence. You want to use a T5 or a transformer model. How do you do? And the simplest is, is to take a textbook on text. So how do people do natural language processing? Well, there are basically several ideas which are possible. One is to say when you have a sequence, so if we think of English, for example, when you have words, you could say, well, a word is a sequence of characters, letters. And so we could decompose it as a sequence of letters, and then that would be the input to the neural network. Now, people have found that this is not very good. I mean, it's a bit debated. Some teams say this is good. Some teams say, it's not very good. One reason for it not being good is that a letter in itself has very little semantics. So when you do that, you expect all the model of the neural network to really, by itself, learn how to aggregate letters to create from individual letters some meaning out of that. And people have found that it's sometimes when you get better results when you decide not to treat a text as a sequence of letters, but when you connect together the letters and create what's called a token. So an extreme token would be to take the full words. So one full word would be one token. But if you do that, then you realize that you have many words which are very similar to each other that become two different tokens. So it's a bit stupid to not to tell the model that faster and fastest have something in common. So there is often, so the current state of the art, if you take the current language models, the GPT-3, the T5, etc., they do something called tokenization, which is more in the middle of this picture, which is to learn automatically a way to gather letters together in small groups. So this is called tokens, with the hope that individual tokens have some meaning. So when you put them in a neural network, for example, at the first layer, they are embedded, they are transformed into vectors, and the vector could represent something about the token meaning. And in particular, on this example, you see that faster and fastest would share one token. So this will help the model know that when there is a sentence with faster or faster, there are something in common about the speed. All right, so this is basically what's done in NLP, and I mean maybe in a few years we will change, but currently it's a bit something that everyone is using because it has been shown to be effective when you want to in terms of downstream applications. All right, so the question is, well, when you have a sequence of DNA like this one, ATT, ATG, blah, 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 what do you do? An idea would be to take individual letters. Taking words seems to be a bit harder because there is no obvious words. So how do you tokenize that? Maybe you can use the same tokenization as the one is used in NLP, but I mean, there is no good reason why it should work. So people in the field of biology have been looking at that a bit. And I think, contrary to what's done in NLP, the most classic, I mean, the most widely used way to tokenize a sequence is to extract, I'll go through it for steps, but it's typically to extract sequences of contiguous letters, but typically with overlap, because it's not clear that you can really decompose a sequence as a sequence of sub-tokens. Often we don't know where they are, so this is not a new idea, but many, I think the state of the art in DNA and protein is more to have overlapping k-mers. K-mers being the length of the typical thing. Like here, you have a DNA sequence, and you see when you extract the 3 mers for k equals 3. The first one is AAG, the second one will be AG6, etc. And that's one description of the k-mer. So if you keep the order, it's really a sequence of k-mers. If you forget the order, it's a bag of k-mers to describe the sequence. All right, so what can you do with that? So here I will go through a series of extensions of the model, but the most basic model is to even forget about the ordering of the k-mers and just represent a DNA sequence or protein sequence as a bag of k-mers. So then you can count how many times each k-mer occurs, and you have a vector representation of a sequence as just an integer vector, and then you can start doing shallow or deep learning on that. Once you have a vector, then you can do linear models, nonlinear models, etc. And in fact, so this is what I go back a few years back. This is one paper that we did, but there are other papers. I think for the people like me who worked on support vector machines many years ago, there was something called the Spectrum Kernel, which is exactly doing that. The Spectrum Kernel is just a kernel for sequences that makes a linear model over the vector of k-pairs. All right, so we, with some colleagues of mine, we also worked on that in some applications in metagenomics. I will not detail the application, but here, just in 2 words, what you want to do is, as input, you have some DNA sequence. So this is typically some DNA that is sequenced, so extracted from some environment. You can look at the soil or the ocean water or the Tokyo subway. You collect some sample, you sequence it, you get plenty of short DNA sequences. And one question is, when you see these DNA sequences, can you tell us which species they come from? Is it a bacteria? Is it a virus? Is it something else? Is it a human, etc.? So this can be phrased as a machine learning problem with kind of plenty of classes. Number of classes would be the number of species you want to predict. So it's typically order of a few thousands or tens of thousands. And the input is a DNA sequence. So we and others have tried to apply this, even a simple linear model on this vector representation, where each sequence is replaced as a vector of counts. You then do a linear model. When you do a linear model in n classes, you have the number of parameters in this model is the dimension times the number of classes. And you can show that it works quite well. And the important thing, so here we had this specific paper I refer to here was at the time an attempt to have large k, so to do k-mers with k as large as possible, to see how the performance was going. And for that, because the size of the model increases fast, we use some, at the time, large-scale machine learning model called Vopal-Wabbit, which was state-of-the-art. And we were able to show that increasing k, so having larger k-mer works well, but we were limited to 12 mers for memory reasons, just because the size is 4 to the 12 multiplied by number of classes. And even if you're a bit smart to compress that, it's still big. Okay, so based on that, next natural idea, and again, the papers have been published over the years, following a bit the trends in deep learning, where you can think of this one as one-hot encoding of the input k-mers. Then this fast DNA model that we worked with on with Romain Menego is more similar to word-to-vec kind of models, where we say, well, we still use many k-mers, but each k-mer we first run an embedding in low dimension, let's say in 50 dimensions, and then we make a linear model in 50 dimensions. So you can think of it as a deep learning model with 2 layers instead of 1. It's not a linear model. In terms of model, it's a way to compress the size of the representation. And doing that, you see that in terms of number of parameters, size of the model, if you have a dimension where you embed your many k-mers, then the number of parameters you need is the embedding matrix. So it's the number of k-mers times dimension. And then the linear model is dimension times number of classes. So you have less parameters than in the previous one. And we were able to scale it to a bit. The problem is that it scales exponentially in k. So as soon as you gain a bit, you cannot still go much further. But with that, we went up to k equal 14. And I skip a bit the details of the results, but basically show that when you do this, so fast DNA, meaning a 2-layer linear model with 14 mers, then we get consistently better results than with 12 in particular in terms of robustness, if you had noise, etc. So it was positive, but we are still stuck at k equal 14 because of memory, because again, you need to start the embedding matrix, which is 4 to the 14 times dimension, which typically is the order of 50. Right, so this is the bottleneck here in this model. It's the embedding layer, which says that if you have k-mers, each k-mer as a vector representation, basically you need to store number of k-mers times dimension numbers just for the first layer, which is how you represent each token, in our case, the k-mers as vectors, before being able to do something after that. And so we thought about, can we go, suppose we want to explore larger k's. This means we want to move from 4 to the 14 to 4 to the 15 or 4 to the 16, etc. So to expand a lot the number of k-mers, what can we do? And a natural idea is to say, well, maybe instead of having one vector for each k-mer, we can start clustering or doing something to say that several k-mers maybe can share parameters, so may share embeddings. And so try to be smart in terms of how do you cluster the k-mers together and just run one embedding for a group of k-mers so that maybe you can trade off the total number of k-mers with the total number of groups that you allow and the dimension. And this is where we started looking at something which is very common in bioinformatics. Actually, it's a concept widely used for genome compression, for example, or genome search. It's the idea of what's called the Brunt graph, but long story short, it's just a way to efficiently see which k-mers occur uniquely in a data set or occur only together. Because, for example, if we take this example here of two sequences, so you have a blue sequence and an orange sequence, you see that you could extract all the k-mers they contain. So here, each small box is a k-mer, so you can count them, and you would say, well, if this was my training example, I would need maybe not to store all the k-mers in memory, but at least to store all the k-mers that are in this picture. These are all the k-mers which are in the database. But when you look at that, you see that, in fact, for example, if you look at the k-mer CAG and AGG, so the first two boxes on the top of this loop here, you see that these are two k-mers, but they just occur in the same sequence. So when you think in terms of training a machine learning model, if you said, for example, the blue sequence is of class 1 and the orange sequence is of class 2, then the two k-mers CAG and AGG, which occur once but in the same sequence, have no reason to contribute differently to the machine learning problem, because they will always be together in a training example or always be absent together. So these are good candidates to be merged together, even though there are two k-mers. You could say I just need to learn one embedding for them because they will just contribute together. And so the double graph is one way to do that systematically, not only on two sequences, but you can, if you have a training set of millions of sequences or billions of sequences, you can build this double graph for all your training set, and then automatically compress it. It's called compacted double graph, just by merging together the k-mers which occur always together in the sequences. And so this gives a sense of how to combine k-mers together, say that we merge together all the k-mers which always occur together or not together in one sequence. And by the way, we also do not, if some k-mers do not occur in the database, they are absent from that graph. So it's also a way to say we do not care about the k-mers which are not in the training set. Right, so basically this is the idea we explore to say, well, when we train a model on a big database, instead of storing the embedding matrix in memory of all possible k-mers per dimension, we will just run one embedding for each of these boxes here, which in this field is called the unity, but think of it as a token. So each time you have a k-mer, it's first mapped to the box where it belongs to here. And then for each box, we have one vector representation. That is the representation we take for the k-mer, and that one is learned in the learning process. Right, so in terms of graph, this means that when we have an input sequence, and by the way, the input sequence then could be at test time, could be a sequence not used at training time to build a double graph. So there may be k-mers which were not seen at training time. But what we do is that we extract the k-mers, then each of them we map it to this double graph to so here we call that the contig. So the contig is one of the boxes of the double graph, which was after compaction. If the k-mer was not present in the double graph, it's back to 0. And then each of them as a vector embedding, and we can merge them together, or we can make sequence model of that in order to have a model for sequences. All right, and so when we do that, we observe that first, now it's possible, there is no more limit on k, because you can think, if you think of the size of the double graph, you can show that it has a maximum value and then it decreases in size. Basically, it finishes with the number of unique k-mers. And luckily, or at least we were happy to see that the performance keeps increasing. Again, I don't go to the details of what we measure, but there is some significant increase when we move from 14, which was the previous best we could explore, to 21, 25, 31. So now the Ks are already much bigger. Now there is a price to pay, which is that we see that it's much better to have longer k-mers. But the longer the k-mer, the more likely it is that you may have a new test sequence, not in your training set, with k-mers which were absent from the training set. Because basically when k becomes too large, the k-mer becomes very specific to the sequences you have. So here we found some trade-off, but I think this is one of the fundamental trade-offs here, which is that as long as you remain in the space of your training sequences, then experimentally having very long k-mers, 30 for example, is beneficial for accuracy. But then you have the risk that a test sequence, as soon as you have one sequencing error, for example, which happens, then the k-mer is absent from your trains. So probably a solution for that maybe to have some fuzzy allocation of k-mers, or a way to cluster not only the k-mers we have seen, but other k-mers where they will be mapped. This mental evaluation. Yeah, you have a question?
So
Yeah.
when k is large, then so if you take the sample in the sliding window way,
Yeah.
then I think overlap is quite heavy. Then doesn't it kind of break the independence assumption of the samples?
Yeah, probably. Yeah. That's right. That's right.
Maybe so when you simply increase k, then up to some point maybe the performance is improved. But after that, samples are more dependent, then like linear classification does not work anymore. Yeah.
So I mean, empirically, we at least so in our experiments, which is only in metagenomics and only something, what we observe is that as long as the test sequences contain k-mers which were in the training set, the performance always increases. But we had more and more sequences where all the k-mers were absent from the training set. So we did not observe what we described, like if the test sequences have k-mers which are present, then for us it was always better. The performance always increased. But I agree with you that because we have a sliding, moving window with overlap, there is no more independence. Except that if I come back, you see to these graphs, imagine instead of 3 we have 31. Then in many cases, when I take the double graph for 31, many 31 mers will be compacted into a unique node, which will be maybe 60 or 100 letter long. And by the way, this is why after a while, all the k-mers become unique, because if each box is 31 and they are merged because of the training set, then a given box may just represent a sequence of length 100 or 200 and becomes very, very unique to the training sequence.
What are the computation time of this debris graph?
I think I'm not sure. So I think it's linear time in the size of the training data set, and not only is it linear time, but many groups are working on making it super efficient. For example, Gunnar Rech is working on that, and many people in bioinformatics. So
does it grow exponentially when k is increased?
With k, no, I don't think so. No.
Maybe not.
No, maybe not. No, because something interesting is that the size of the double graph is not really dependent on k. The space of k-mers. So you can think of the double graph as a very sparse subset of all the k-mers, but it just builds a k. It just reads the training sequence and counts the k-mers. So I'm not super sure about that. Maybe someone in the audience knows better than me, but there is a specific literature on that, and it's super.
Okay.
All right, so but basically this is what we did, and I think there are plenty of other questions. We discussed a few of them, but some ideas that we discussed and discussed even with colleagues but never did were, for example, here I said that we use a double graph to define the tokens, and then we learn an embedding of each token. But whoever in the room has heard the word graph neural network may say, well, what we're doing at the end is embedding the nodes of a graph, a very specific one. And we don't use anything, we don't use any technique. We did not use any technique related to convolution of the graph, etc. We just say, well, the graph defines tokens, then we forget about that, and we run an embedding for each token. So I think there could be some interesting direction to check if the graph structure could penalize or constrain a bit the embedding of the tokens. Another idea is to say that at the end of the day, when we use this specific graph, then a sequence is just a path over the graph. You can really represent each sequence as a path over the full graph. So when we think of, what we want to do at the end of the day is machine learning over a path of a graph, and maybe there are other ideas that we could use. Here I just mentioned it's a path of a graph, but I just say it's a set of tokens, a set of nodes. So plenty of things could be imagined here. And another idea that we started exploring, but again, it didn't go anywhere yet, is learning to tokenize. So I said that NLP is using one tokenization, bioinformatics uses overlapping k-mers, but all of them are like, let's do that, and then independently, once we do that, learn the embedding, etc. It would seem interesting to say, well, tokenization may be part of the learning system, and we may want to learn how to tokenize a sequence as part of the global optimization. Okay, so I realize I spend lots of time on this sequence, which is supposed to be just the appetizer of the talk. But that's okay, because I have as long as I want, I think so. So maybe we keep the questions even for the end, and we'll continue with the second part. So second part is a bit specific. Maybe I will be a bit quicker because I think there is nice mathematics, but maybe it's just to illustrate to you how some constraint on the field can be analyzed and can be derived into constraint of some model. So this part is joint work with a PhD student of mine, Vincent Mallet from the Pasteur Institute, and was presented at NeurIPS 21. Okay, so as I said in the introduction, here what we investigated is the fact that when people say a DNA is a sequence, for people who know what DNA is, it's not exactly a sequence. DNA is a molecule. Here is a zoom over a picture presentation of the molecule, and you know that DNA is a double-stranded polymer. That we have a convention to call each block. So each block is a nucleotide. There are 4 nucleotides, so we call them A, C, G, T. And probably you've heard or you remember that on the 2 strands, in fact, you don't have the same nucleotides because they have to match each other. So each time you have an A on one strand, then you have a T on the other one. And each time you have a C, then you have a G. This is the biology, right? So in the picture on the left, each time you have a blue, then you have a red on the other one. And each time you have a violet, a purple one, then you have some yellowish on the other one. So this means that suppose you have a machine learning problem, which is you extract DNA. This is what we do in metagenomics. I said when you sample the Tokyo subway, you extract DNA, which could be double-stranded, and then you sequence it. And so when you sequence it, in fact, you extract just one sequence of ACGT, which can be one strand or the other. But the choice of the strand is completely arbitrary. There is complete symmetry, or anti-symmetry, meaning that when you sequence, you always sequence in what's called from 5' to 3', meaning if you have one strand, it's from left to right, and if you have the other strand, it's from right to left. Right, so this is because of biology. And so this means that, long story short, when you're interested in one part of DNA, there are basically 2 ways to represent it as a sequence, which are illustrated on the upper right picture here. Either it's a sequence on the top, which you read from left to right, so it's A, T, G, C, blah, blah, blah. Or is the sequence in the bottom that you read from right to left, which is then A, T, G, etc. And so when you have these 2 sequences, you have to remember that they represent exactly the same molecule. Right? It's just a fact. So usually when we sequence, we forget about that. We just say, well, we sequence this thing. Or I must say that if you're not careful, what biologists like to do as well, that because they're aware of that symmetry, sometimes there is a convention to say, well, if you sequence, if you get a sequence, then you compare this sequence to the reverse complement. So this is the second sequence, and you just decide to keep the smallest one when you take the lexicographic order. So it's a way to map any sequence either to itself or to its reverse complement in a systematic way. Okay, but this is something, this is very different from English or Japanese. In Japanese, you take a sequence. Well, you cannot say the meaning of this sequence, of this sentence could be written the same way with some other sequence of characters, which are completely different and read from right to left, etc. But this is true in biology. So why do we care about that? Well, we care about that because maybe there are some invariants we want when we suppose we have a deep learning model for DNA. So as input, you would have the DNA sequence, and as output, you would have something. So it could be either a classification of the sequence, or it could be even a classification across the positions. Like, you have a DNA sequence, and for each position, you want to predict something. That could be the secondary structure or the property of each position. Then taking the example of, suppose you want to map a signal, then if you look at this plot, which is probably not a very good plot, but imagine that you have a DNA. So the long DNA is at the bottom. This is, the genome is very long. Then when we sequence a part, we get a subset. So each arrow here, think of it as a text, ACGT text, that describes the DNA between position A and position B, either on the on the left to right strand when it's red, or on the right to left strand when it's blue. Okay, so you have to think of this. So each arrow here would be one sequence of ACGT, and we know that suppose you want to to make a machine learning model that predicts a property of DNA, maybe we want to predict this black time series on the top, which is just a measure that you want to predict at each position. And if we use a machine learning model, it would be nice to say that whatever the input arrow you have, so the input text you have, maybe you would like the predictions to be compatible with each other, meaning in particular that if you translate a bit your input, then you would like the output to be translated as well, so that you would describe what's the, independently of where you sequence, what's the correct output. And more importantly, if as input you have the left to right strand or the right to left strand, you would like to have two predictions, which also would be the same up to the right to left, left to right symmetry, right? Because that would be the signal of the DNA. So that's a constraint that we may want to input a neural network, which is how can we have an architecture, a model that ensures that we are compatible with these two symmetries, translation and reverse complement. Actually, some people have talked about that in bioinformatics. Here are a list of a few papers, and in particular, one paper presents something called, they named it RCPS, it's called the Reverse Complement Parameter Sharing. So it's a convolutional neural network. I'll skip the details, but at the input on the bottom, you have a sequence of ACGT, which is one hot encoded. And then you have several layers. These are convolutional layers. You have nonlinearities in between, etc., to produce on the top some output. And they solve the problem of being invariant by translation and reverse complement by adding some constraint on the parameters. This is what they call the parameter sharing, the PS. And here it's just summarized through the arrows. So it's something non-trivial, but when you think of it, I'm sure you can find it. But basically they say that if you have an A on the left, so if you have some weight that applies to an A on the left, then by reverse complement, you would like to have the same weight applied to the reverse complement, so to a T on the right of the sequence, this kind of thing. This is the notion of reverse complement. So change of letter and change of position. If you think of it, then you can enforce that the parameters are the same, and therefore that by design, when you have as input a DNA sequence or its reverse complement, then you get two predictions which are symmetric to each other by the horizontal symmetry. Okay, so this is, I found it very nice, and the question we addressed is that the only architecture that's possible, or can we characterize are there other ways to do that, right? Mathematically, can we study the constraints it imposes on the neural network to ensure that the symmetry is taken into account? And so this is where I think, for the sake of time, I will go a bit fast. I will just illustrate the results we got. But basically, the good news, what we found interesting is that we can translate this question into a very well-defined mathematical question because, because the two things that we discussed, meaning you have a DNA strand, and maybe you can look it a bit on the left or on the right, so this translation sampling, and the reverse complement sampling can be mathematically defined as some group action. So group in the sense of algebra group. Group is a set with things that you can compose. And here we, if, for example, if we define a DNA, so an infinite DNA sequence as a mapping from Z to, for indices that describe A, C, G, T, then and by convention you say that the text I have is the sequence between position 0 and L. This is what you see. Then translating means that you sample your window on a translated signal. So this means that your function f that describes the DNA is translated. So it's very classical. Translation by T means that instead of f of x, you look at f of x minus t. This is translation. And on the second one, which is reverse complement, is a bit more subtle because you need to inverse the right to left or left to right, and you need to change A to T and T to A, C to G, G to A. So this can also be seen as a group action. Now it's a very simple group, which is just a group -1, 1. So meaning either you do nothing or you do a reverse complement. And if you do it twice, you come back to the original one. And mathematically, if S is either 1 or -1, then what you do is you take your signal f at a position which may or may not be inverted. So S becomes you invert the indices. Plus you multiply f is a vector, you multiply by a matrix to permute the letters you have. So sigma of S here is a matrix, a permutation matrix that tells you how the things are changed in terms of vector. Okay, so it's a bit technical, but long story short, here we have a group which is defined by T, which is an integer that describes how you translate, and S, which is -1 and 1, which tells you whether you take which trend you take, basically, whether you take this trend or not. And once you have a group, then it's possible to express the invariance we want in terms of a diagram. And maybe this is the only, so it becomes a bit more abstract, but this is the translation of what I said earlier in terms of, if we translate or reverse complement, we would like the output to be translated or inverted right to left. This can be translated exactly as saying that if we want an operation, so phi will be the operation we want, that transforms some input to some output. So this could be between two layers if you want. In such a way that, so the input would be, think of it as a DNA sequence. If you apply a given transformation defined by a translation T and a possible reverse complement S, you obtain a different signal as input, right, on which you could apply your model. And what you would want is that when you compare the prediction on the Ross sequence or the transform sequence, so phi on the left and phi on the right, you would like both of them to be compatible with each other, meaning typically you want some right-left inversion, and potentially if the output is multidimensional, you would like as well the equivalent of the A to T, C to G conversion that would apply to this signal. So potentially, like if you have a two-dimensional output, maybe in reverse complement, you would like to reverse complement. Okay, but so this means that what we want can be expressed in a simple equation at the bottom, which is that phi, which is our unknown. So to be compatible means that when you apply phi to, so pi2 is some operation that you apply to the phi of the input. This should be the same as phi of pi1, which is applied to the input. Right? This commutativity is the mathematical translation of enforcing the constraint. Okay, so it becomes abstract, but the good news is that now, and maybe some people in the audience may have seen that before, this thing has been studied by many. If you've heard some talk of Taco Cohen or Maxwelling or other people in the field of geometric deep learning, is the topic of equivariant neural networks, because this thing is exactly a notion of equivariance. So we don't want invariance, we want an equivariance, meaning when a group acts on some data, and usually, so, if you go to NeurIPS, usually it's not on DNA, it's on images on the sphere. Things like, suppose you have an image as input, if you rotate or translate the image, you get a representation, and you would like the representation to be the translated or rotated image of the representation of the translated image, right? Same on the sphere. So, it's interesting because this can be studied mathematically, and there is a very clear description of what constraints it puts on phi. So here it's psi, but if you want to have this equivariance, what can psi be? And so this is where, finally, as promised, I will skip a bit. But basically, we can answer very rigorously the types of mappings files, so the layers that can be used in neural networks for DNA that are equivalent to translation and reverse complement. Right? It's just a question of studying the group of translation times reverse complement, which is quite simple mathematically. And so there are a few theorems. Maybe I will just skip that one and mention that what we can show is that in terms of one layer, thanks to the translation variant we want, we can show, which is not a surprise, that it has, if it's a linear layer, it has to be a convolution. So among all linear layers, only convolution are allowed. And in order to be robust to reverse complement, then it's a convolution with the kernel that has to satisfy some properties about some alpha beta that have to be even and odd. So this can be clarified. And in particular, so the previous architecture I mentioned, the RCPS, is a particular case of that, but it's not the most general one. It's a particular case of that with some specific dimensions and specific choices of matrices. Right. So this is, let's say, the complete generalization. Now we can also discuss what non-linearities are possible. And again, it's possible to study it. So, I mean, the theorem is a bit ugly, but it's complete. It describes exactly all the non-linearities that can be allowed, depending on the layers we have. And again, the RCPS was one particular example of that, but there are many more examples of that. So at least we can characterize all. Okay, so this is the first result. So just starting from this thing that we want to enforce, now we can fully describe it, express it mathematically, and from the mathematical expression, obtain some theorems that describe the layers and non-linearities which are allowed. And then the question is, does it work? Is it useful to do that? Maybe I will just focus on the upper right plot on this slide, which is basically the performance on some tasks. So here the tasks are, you have as input a DNA sequence, and you want to predict a signal, which is how well a given protein will bind to the DNA. This can be measured experimentally, and we believe that it should be possible to predict the intensity of the binding from the sequence. So it's a DNA sequence to continuous signal prediction problem. So we try that with different architectures which are convolutional neural networks. But here there are 3 plots. So there are 4 plots, but these are just different proteins. So we can just look at the right one, which is the average. Overall is the average of several tasks. And there are 3 models. So the dark blue one, which is not is good, but the worst one is a standard CNN that is just a CNN. Then the middle one is the previous RCPS architecture. So we will confirm what they did in their paper, which is that you gain performance by using this, incorporating the symmetry in the architecture. And the red one is our new equivariance, so basically which is obtained by a grid search over the space of possible parameters that we described. Of course, it's not overfitted, train, validation, tests, etc. But it shows that there is still some gain to be obtained by exploring a bit more of the space of equivalent models beyond the one that was proposed earlier. Right, so I mean, it's maybe it's not a revolution for biology, but at least it shows that it's a specific problem. There is some magical constraints, and if you incorporate them in the model, there is hope that you change a bit the model and you get better prediction.
Question?
Yes.
So when you impose equivariant, so in implementation, so how did you really make it? What's the computation over?
So the computation, basically the computation is no more. It's just that it imposes constraints on the parameters. So it's a question of parameter sharing, if you wish. So it's quite, so the light blue is also parameter sharing. It says, you do a conversion with you have parameters of the conversion kernel, and parameter sharing just say that some parameters are equal to other parameters. So you have less parameters actually to fit. And so when we extend it from the light blue to the red, it means that we have more possibilities of parameter sharing, but it's always parameter sharing. Yes. So by the way, there is a GitHub link here. If whoever wants to check the details of it's implemented.
And the parameter sharing structure is given in advance.
Yeah. You can think of it as basically it's the same architecture as the CNN, but we have less parameters. So in particular, actually some people say that equivalent neural networks, etc., it's beautiful but not very useful, because if you have infinite number of data, then you don't gain anything by restraining yourself to that. I think here we are in situations where the performance is not perfect yet, and if you don't have infinite number of data, then you can think of it as having less parameters. So in terms of training, it's a bit faster actually.
Yeah, that's good.
All right, so this was the part about the architecture, and I will conclude the talk with something we did recently that involves differentiable programming on tasks, so on loss functions, which are a bit specific to proteins and sequences. So in the field of bioinformatics, probably if you take a course on bioinformatics, the first course is about sequence alignment. This is really the important thing in bioinformatics. Sequence alignment is the task when you have two or more sequences to try to align them together. And basically that's the way that people believe and know it's good to compare sequences, just because most sequences which are related share something in history. Like in the past, probably they were just one sequence in one organism, and then it got split and given to the.
I think the video is not updated. Okay.
Thank you. Yeah. So I was talking about that. So for example, for SARS, people have kept sequencing the SARS-CoV. So either the one in humans or the one between different species, in order to understand where it comes from, etc. And all the things, they are just sequences, but they are aligned together in order to capture positions which are conserved, positions that mutate, etc. And by the way, what I mean by alignment is that if you look closely here, maybe it's too small, but horizontally you have different DNA. So these are protein sequences, right? You have some V, some. Sorry, this may be DNA. But the important thing is that sometimes you have a dot, and a dot means that there is no letter here. Like the raw sequence is a bit shorter, but the alignment is to say, even though the sequence is shorter, we should insert something here so that the rest of the letters seem to be aligned. And probably in the phylogeny, so in the past, there was a letter here which disappeared by mutation. Okay, and so sequence alignment is important, and the first thing you learn in bioinformatics 101 is how do you do alignment. And this is it's one of the one moments when you do bioinformatics, is that looking for the best alignment is hard, but you can express, if you define a specific objective function, you can express it in a way that can be solved by dynamic programming. So it allows you to find the best alignment between two sequences by solving in quadratic time this quadratic programming program that finds the best alignment. If you're familiar with time series, this is the same as dynamic time warping. How do you best align two time series? And it's a way to align sequences. Now, it turns out that this aligning sequences is good, but so this dynamic programming maximizes some score function over all possible alignments. And the score function itself depends on parameters, which are typically fixed. You have parameters called substitution matrix, gap opening, gap extent penalties. So these are parameters that you fix. And when you fix them, you run the dynamic programming to get the best alignment. But if you change them, you get different alignments. Right? So a question is, well, so suppose we want to improve how we do alignment now. It's not that, for example, alignment is not perfect. So the algorithm always returns something, but it has been shown that if you have two sequences which are kind of different, even though they used to be the same in the past, it can be hard to recover the correct alignment. So the question is, how can we improve how we do alignment? How can we maybe better choose the parameters so that the algorithm finds a better alignment? Right, so that's the case. For example, these are, most people here are not biologists. So for biologists, this sequence probably makes sense. But here, these are two sequences of a protein. one in a gorilla and one in a mallard. I think mallard is a type of duck, which in fact are the same proteins because they have been studied. We know that they are the same 3D structure, they do the same thing. So they have a common ancestor. But if you look at the sequence itself, it's very, very hard to see any similarity here. And in fact, this is the correct alignment that is given because we know the proteins, we know the structure, etc. And if you look closely, so there are a few gaps, and there are just a few letters which are conserved. So you have CC, CC, then VV, and all the other letters are different. So this is an extreme case of very, very hard alignment problem, which if you are just given the two sequences, typically you get, if you run your dynamic programming, it will not find, it would find that one. This is state-of-the-art aligner. It produces some alignment, which is completely wrong. Okay, so the problem is difficult in these changing cases. Okay, so we worked on that, and we did that with a group of colleagues at Google. So this is just a one-slide summary of a long paper, but what we did is we said, well, let's try to borrow these ideas of deep learning for sequences, but incorporate the task, which is learning to align at the end. And so we proposed a model. Maybe if we just look at the top of this picture here, a model that produces an alignment and hopefully that produces a good alignment. And the way it does it is it starts from two sequences on the left. So here we have two short sequences. The two sequences are mapped to a continuous space by a language model. So this could be one of the models I've described previously, or it could be any transformer type of model. It's parameterized. It produces a sequence of alignment. And then what we propose, this is the new thing, is that when you have two sequences that are mapped to two sequences of vectors by some deep learning model, then maybe it's possible from these two sequences of vectors to create some parameter matrices. So S, E, and O would be substitution, gap opening, gap extension, that then could be the input to the Smith-Waterman algorithm, so the dynamic programming model that produces some alignment. Okay, so here conceptually what we want to say is that in order to align two sequences, we will apply Smith-Waterman, so we will apply the dynamic programming, but the parameters of the dynamic programming would be the output of some model here that combine deep learning with some. So the P model is just a way to from two sets of vectors to produce matrices, which are parameterized. And hopefully, we may be able to learn the parameters by learning to align correctly, a training set of proteins for which we know the true alignment. Okay. But so the use is just that. So at test time, we just embed the two sequences and then compute Smith-Waterman. And in order to train the model, then suppose you start from a set of pairs of sequences with correct alignment. Then what you would like to do is, you take your two sequences, you map them, you compute the alignment. It may be correct or not, so you would like to backpropagate some objective function, to change the parameters anywhere so that the score matrices, so S, E, and O, are changed a bit in your direction to improve the alignment. Which raises the question of how do you make this differentiable? Because the Smith-Waterman produces a discrete alignment and it's not differentiable. If you change a bit the parameters, it doesn't change the alignment. And so this is where there could be a full talk about how you make differentiable discrete operations. I will skip it. I'm happy to discuss it further. But for those of you more interested in these aspects, we worked a lot with again colleagues at Google and others on either techniques based on regularization or perturbation. So the end goal of that is to say that many problems, and alignment is one of them, can be seen as the argmin of an objective function. So, the dynamic programming, in fact, solves a linear problem. It says what is the alignment that maximizes a score, which is a linear score over the alignment. So you can think of it in some space as a subset that is the space of alignment matrices. What we do with the dynamic programming is we maximize a linear problem over that polytope. So we usually find just one vertex of that polytope, which is an alignment. And the question is, how do you make this army differentiable. And so by regularization of perturbation, we're able to define a notion of soft alignment, if you wish, just like the soft max is different from the max, which then it becomes differentiable and so allows to compute a loss that could be backpropagated.
So the idea here is to substitute the problem with a relaxation in a continuous space.
Exactly, yeah. And so when we do that, then we can solve this problem of you have correct alignment, you have differentiable prediction, and then you can define a loss that can be backpropagated, and then allow to train all the parameters of the model so that it learns to produce matrices that then give a good alignment. And the good news, so I will skip a bit, but good news is it works well. It works well. So we tested its capacity to do either to detect when two sequences can be aligned or not. So it's a question of homology detection. And here, when we compare, so blue is state of the art, orange is the new one. When we have difficult cases on the left, horizontal axis is the difficulty of the problem. Like on the right, it's very easy to recognize. On the left, it's very hard. So it's not perfect, but we have a big boost in recognizing remote homologs. And the same, so here it's still very far from being perfect, but we move from very, very bad to slightly better on the very hard cases. And so all these curves are obviously on strict cross-validation, where the test set are very different from the training set as well. But it seems to be learning something at least. So the idea to combine this differentiable loss function with learning to embed proteins seems to have some good impact here. And I will conclude with some other application, but maybe I will just keep that. If anyone is interested by how this helps to do DNA sequencing, then please ask me questions. In just one sentence, we did collaborate with a company called Pacific Bioscience. And for whoever those genomics, this is super famous, PacBio. They produce a machine that sequences DNA, but the machine produces DNA with a lot of errors. And so they're very interested to complement what they produce by some algorithm to decrypt their signal in the correct DNA sequence. And for that, in fact, they use deep learning. So they translate their signal into a matrix of data that describes the experimental measures they do. And the goal is from all these measures to produce a DNA sequence at the end. That would be just the DNA which is sequenced. So it's very machine to translate the signal into DNA. And so in this machine, there is in this method, there is in particular some deep learning model here that they were doing, but they had difficulty because they had to compare a prediction. So a prediction for them would be a DNA sequence they believe is the true one. And this is to be compared with the actual one that we may know if we do control experiments. So they needed a loss function to compare a predicted DNA sequence with a true one. And typically the predicted one could be shorter or longer, so it suffers from the same things that differentiate the gorilla and mallard sequences. And so here we work with them to incorporate a differentiable alignment loss. So again, it's the idea to say maybe we can align your prediction to the true one and then make it differentiable so that you can optimize the weights of the model. And they were quite happy because they really improved the accuracy of their models. They do basically 40% less errors in their prediction. So it's a significant, just with the algorithm, they don't change the machine, but now they can sell a sequencer which is 40% more accurate. So they were very happy about that. And this is going to be published on Nature Biotechnology very soon. Okay, I'll stop here. I'm sorry, I really was too long, but you should not have told me I had as much as I wanted. So, basically what I wanted to say is I think it's very promising to use deep learning and in biology, but we are decided to go from the discrete space of sequences to continuous model and then doing some fancy things. We can learn a lot and borrow a lot from natural language processing, but I try to show you that sometimes I'm sure there are many other possibilities to have dedicated efforts to biology. It could be symmetries, it could be the tokenization, it could be other things. And of course, I did not talk much about the applications, but there are lots of exciting applications. You may have seen and heard of AlphaFold, which is really a revolution in biology. That is a sequence to 3D structure model. There's there's lots of exciting model recently on language models that allow, for example, to predict the effect of mutations, either the effect on the structure, on the phenotypic effect, etc. So people are super excited about that, and I think this could be, if some younger scientists, students are in the room, I think these are cool topics of research. Thank you for your time, and if you want to ask questions, I'm happy to take some.
I have.
Yeah.
Not a student.
Almost. I mean, I'm a young.
Yeah, can you open
I'm a young researcher, and you are.
Can you open your camera? So.
Oh yeah,
yeah.
That's great. People can see you.
Yeah, so I want to ask a question about the differentiable, extension part.
Yeah.
Because it's a kind of traditional story, right? When we have a zero-one loss, we wanted to relax it into hinge loss. When we have a clustering problem, we relaxed it to k objective. So here, what was the kind of advantage of using this differentiable alignment? Or any guarantee?
So I don't know if there's a specific advantage. At least it's I think it's the first time we have a differentiable notion of alignment. So I would say actually the idea that we use are very general and they are not new. So for example, when I talk of regularization, this is exactly what's done with the softmax. People know that what is the max. Now the softmax is a proxy for, in fact, the argmax. Right? The argmax would be, this is the largest item on K.
Annealing, assimilated annealing.
Exactly. So it's possible to phrase it as some exponential blah 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 normalized. And so if you see the max geometrically, then you can think of it as the argmax is one point of a specific polytope, which is a simplex, and the softmax is one obtained by regularization, entropic regularization. And so basically we use the same idea, but we just said, well, this idea can work with any polytope. So as soon as you have, this thing works for a linear optimization over a polytope. And here what we did is just to realize, which is not very new either, that the alignment is linear optimization over a specific polytope. But the idea itself is used in softmax and is used, for example, in the regularized optimal transforms. It's entropic regularization, so the synchron method, etc. It's exactly the same idea on a different polytope. Right. So here I wouldn't say that we have a new method. We could have some guarantees, etc. But here the novelty is more to apply it to a specific polytope, which is alignment. Now the second method, which is perturb, I think it's a bit less used, even though it's also well known. It's called the Gumbel tricks when you want to approximate the argmax by a different way. So people, when they focus on max, they can take the softmax or the Gumbel tricks. These are two ways to sample the maximum thing. So here again, the perturb trick is not novel, but we have this paper with Cotin and Francis Bach and others that narrate that in particular has some more general theorems about what it solves, what's the trade-off between approximation and accuracy. Sorry.
Do you have any hyperparameter like temperature?
Yes. So in both cases there is a parameter. Yeah.
You
Exactly.
can schedule that parameter in a nice way to better than.
So I think conceptually it's possible. We did not do that, but yeah. So think of the softmax, etc. Indeed, when you have the temperature which is very high, meaning you're very smooth, then it has some properties, and even with the softmax, I'm sure people already include varying the temperature with training the model.
If the temperature is high, then I think given constraints are not really satisfied.
Yeah.
You need to somehow lower the temperature. But if the speed of lowering the temperature is too fast, then maybe the algorithm is.
I agree. So I think it's a good idea. I'm not sure I would know how to do that, but so there is this parameter that probably allows to interpret between a very smooth objective function. So you could think of starting training over the smooth thing and then reducing the smoothness so that you approximate a bit better your loss function.
From a practical viewpoint, I think this kind of continuous reactation is somewhat well-known technique.
Yeah.
previously. But now we can utilize deep learning framework to optimize it in a computational efficient way.
Yeah.
So that's a kind of practical impact here.
Yeah. So I think so in most of our applications, and we see it actually, at Google, many people have discrete optimization. In fact, we have a whole research program, which is really, we call it differentiate everything. So the idea is now that we have all these efficient models for large-scale computational graph, TensorFlow, PyTorch, JAX. What they need is a basic operation that does something in the forward mode, and that can do something in the backward mode, so the backpropagation. And so these ideas are super simple, but as soon as you translate them in new operations that can be backpropagated upon, suddenly they can be inserted in computational graphs, so in deep learning models. And sometimes you can have a model where you have some align layer, some match layer, some many layers, some shortest path layer. We've been working on shortest path problems, so you can differentiate the shortest path. So I think it has a specific interest in deep learning frameworks.
So how does the combinatorial optimization people find this kind of work? Because they always have a nice meta heuristic for each problem, and they have their own implementation for each task.
Yeah. Well, so I don't know. We discussed a bit. I did discuss with some of them, but we did not go very far. I would say that so here, everything I presented and I've been working on recently is the discrete problem is not challenging itself. So like we say, we need to solve an optimization problem over a discrete thing. So this is a bit scary for us, for whoever only knows the gradient descent. But dynamic programming works, shortest path in the graph works, etc. So at least these problems are not the NP-hard problems. And we're not talking of saying you have an NP-hard problem and we propose to solve it with gradient descent. I know some people do that, but I was not going to do that. So all these things are more about you guys have a very efficient algorithm to solve a problem, a discrete problem. Now we can use your implementation to make it differentiable. And I would say in particular, I did not present it in detail, but what we call the perturb thing is a very powerful method for that, because the trick is super simple. It's like the Google softmax tree. So it says that if you have an optimization problem that is, produces a discrete output, but that depends on continuous input, then you can just randomly add some noise in the inputs and use your optimization to obtain some differentiable things. So your new loss is like the expectation of your output under the input noise. And so what's important here is that in implementation, you just need to borrow what the combinatorial guys know how to solve, and just say, well, let me add some noise in the inputs, and I will have the good formula then to obtain the gradient as using your thing. So I think it's an excellent fit actually between discrete optimization and differentiable optimization.
Yeah, thank you. That's very nice.
I have a question.
Yep.
Basically, recently, many researchers found out that deep learning actually have a very inherent problem. For example, you have some perturbation in the input, they will make a huge mistakes.
Mm-hmm.
When you apply the deep learning models into your biometry, for example, DNA sequence, for example, make prediction, you do some of the stress tests for this. For example, if you change slightly of the input, will make a significant difference. For example, the rabbit can become a cat.
Yeah.
In terms of DNA.
Yeah, so I think first that the problem is probably true for many deep learning models, including the ones used in biology, right? That's for sure. Now, I would say it probably depends on the specificity of the model. But for example, all the models that I've shown take as input a sequence. So here we don't really have the possibility or the notion to have like an epsilon modification of the inputs, because the input is discrete.
Yes.
So at least, as I said, we go from discrete to continuous, blah, 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 but let's say an attack or modification of the input would be in the discrete input.
Right.
And I think the models are quite robust to change of later, because they are really trained at the resolution of the possible modification. So I'm not sure there is so much problem just because the input is discrete.
Yeah, but actually we can do some like the replacement attack. For
Yeah.
example, we just replace some tokens.
Yeah.
Then problem check. In ARP problem, there is some
Yeah, it's
some
true.
reason.
Yeah, I think. Yeah, that's a good question. I'm not sure what to answer. So another thing I can mention is, I said that in my conclusion that one of the most, what I think is one of the most exciting topics is predicting the effects of mutations. So what this is typically some people train models when input is a sequence, and then they one by one change each letter in all possible order letters, and the alphabet is very small, and look at the impact on the something. So it could be on the output, it could be on the probability, etc.
Right.
And I mean, this thing seems to be useful. In particular, this thing seems to be a very good predictor of how bad a mutation would be if you did that in the wet lab. The question of fitness, etc. So I'm not sure we are in the regime where it's a weakness of the deep learning model. Like here, so far it seems that the systematic exploration of the neighbors of the input has proven to be correlated with some real biological things. So it's very interesting to better understand that.
Yeah, I guess that would be useful to understand how the deep learning works.
Yeah.
But it's very helpful. Thank you. All right, Prof, do you want to take a photo for our on-site meeting?
Because it's a hybrid mode.
Yeah, it's a hybrid mode, but we have the very first, we can have a very good concept.
But after closing the session.
Yeah, I think so. There are no further questions. We will conclude.
Okay, then so thank you very much, everybody. So please join me to thank Jean-Philippe again. So.